Good morning, boys and girls. Today I am going to put the solutions for the assignment that I given last time. All of you are able to do the cross regarding the simple dihabit cross. The cross which involves the tall pea plant with violet color flower and the dark pea plant with the white color flower. So all of you are able to do that question. So I will be dealing with the next question over here, which shows a cross between a pea plant heterozygous for round and yellow seeds with a pea plant having wrinkled and green seeds. Okay, so most of you have done a mistake over here regarding the selection of alleles for the heterozygous parent having round and yellow seeds. Okay, so I am going to put the solutions for this particular question, and in this particular question only we will also discuss about the test cross which we have learned in yesterday's class. Okay, so basically first of all what you have to learn, you have to check out the parental characters which has been taken for the cross. Okay, so in this particular question the pea plant that has been selected is heterozygous for round and yellow seeds. Okay, so here the parental character which is taken is heterozygous for round and yellow seeds. Heterozygous means where the alleles will be different, and this particular cross is a dihabit cross, which because we have taken two pairs of contrasting characters or four characters. So the first parent is heterozygous for round and yellow seeds, and the second parent that we have taken is wrinkled and green seeds. So over here the round and yellow seeds are dominant character and the wrinkled and green seeds are the recessive character, but the round and yellow seeds are heterozygous. So therefore heterozygous means the alleles will be different. So for round the alleles has to be capital letter R, small letter r. For yellow it has to be capital letter Y and small letter y. So this will be our heterozygous allele. So this one is our heterozygous allele. If the allele is to be capital R, capital R, capital Y, this will be our homozygous allele. But the question says the pea plant is heterozygous for round and yellow seeds. So this one, capital R, capital Y and small y, this is our heterozygous allele. Okay, not this one, this is our homozygous allele. Okay, and next one is wrinkled and green seeds, which are recessive character, and recessive character will be always expressed in homozygous condition. So therefore it will be small r for wrinkled, and for green it will be small y. Okay, so these are our parental alleles. I hope it is clear for you now. Okay, so the question number A says, so the cross in Punnett square. So what we can do, so let's start over here. So we have got capital letter R, small letter r, capital letter Y and small letter y, which is heterozygous for round and yellow seeds being crossed with wrinkled and green seeds. Okay, now let us form the gametes from these parents. So over here you can see the allele is heterozygous, so the gamete in this particular condition will be capital R, capital Y. Okay, first gamete will contain the allele capital R, capital Y. The second gamete will contain the allele capital R, small letter y. This is the second gamete with the allele capital R, small y. The third allele will be small r, capital Y, and the fourth allele will be small r and small y. These are the four types of gametes which are being produced from heterozygous round and yellow seeds. Now from here the alleles are homozygous for wrinkled and green seeds. So since the alleles are homozygous, all the gametes, the four gametes that will be produced from this particular parents will be same. It will be small r and small letter y. So all the four gametes that will be produced in this particular parents contain the allele small r, small y. Okay, now let's put this one in the Punnett square. So when we put this one in the Punnett square, let's check it out. What will be the offspring that will be produced? Okay, so we have got the allele small r small y. I can write it like this way. So the second gamete we can find capital R capital Y. Okay, and we have got capital R small letter y. The third gamete we have got small r capital Y, and the fourth gamete we have got small r small y. These are the four types of gametes that are being produced. Okay, now let us try to find out the progeny or the offspring that will be produced when this gamete will fuse. So here we can write down when this will fuse this one, capital R small r capital Y small letter y. Okay, now this and this, capital R small y small y. Okay, now this gamete with this gamete will be small r capital Y small y, and finally this and this last gamete will be small r small y small y. Okay, so the progeny or the offspring that are produced as the allele capital R capital Y small y, capital R small y small y, small r capital Y small y, and small r small y small I. We just doing it for your understanding only. Okay, so when we look at the phenotype, okay, when we look at the phenotype of this offspring, so what will be the phenotypes? So look over here. So this one is round because capital R is dominant of small r, and capital R means round, so it is round, and capital Y is dominant of small y, capital Y means yellow, so it will be round and yellow seeds. Here same thing, this one means round and this means green, so it is round and green color seeds. Now here small r means what? It means wrinkled, it means wrinkled, and capital Y small y means yellow, so we have got round and yellow seeds. And finally we have got small r means what? Wrinkled, small y means green, and the last one is wrinkled and green. Okay, so therefore second says they write the phenotypes of the progeny. So these are our progeny which are obtained by crossing the heterozygous round and yellow seeds with the wrinkled and green seeds. Now the phenotypes that are produced are mainly round and yellow. Okay, then we have got round and green, we have got wrinkled and yellow, and we have got wrinkled and green. So these are the different types of phenotypes which are produced, okay, when we are crossing the heterozygous round and yellow seeds with the wrinkled and green seeds. Clear this one? Now the next part is, what is the cross known as? So this is question number A, this is question number B. Now question number C says that what is this cross known as? So if you look over here, this capital R capital Y small y means it is an F1 progeny. So here F1 progeny is being crossed with the recessive parent. So in last class only we have learned that whenever we will cross the F1 progeny with the recessive parent, then that particular cross will be known as test cross. Okay, so what is this cross known as? The name of the cross is test cross, and state the purpose of conducting such a cross. Now why are we performing such a cross? To find out the unknown genotype of the F1 offspring. Okay, so purpose of this cross is to find out the unknown genotype of F1 offspring, because we don't know whether the offspring is homozygous dominant or heterozygous dominant, and for that we used to perform the test cross. Okay, now I'll just elaborate a little bit about the test cross. Okay, so in this class we have learned that, look over here, in test cross what we do, so when we normally cross, like let's explain from monohybrid cross, okay, quickly. When we cross a tall pea plant with a dwarf pea plant, then therefore in the F1 progeny the plant will be tall, but we don't know whether the plant is homozygous tall or heterozygous tall. So in order to find out whether the F1 progeny is homozygous tall or heterozygous tall, we will perform which cross? Test cross. And for that what we do, we will cross the F1 progeny with the recessive parent. Like we have done two crosses, one is back cross and second one is test cross. In back cross what we do, we will cross the F1 progeny with either of the two parents, with the dominant parent or with the dwarf parent. So when we cross the F1 hybrid with the dominant parent, then therefore all the progeny, okay, in the offspring will be tall. Look over here, capital T, capital T, capital T, capital T, capital T, small t, capital T, small t. That means all the progeny will be tall when we cross the F1 progeny with the dominant parent. But in the F2 generation we have obtained the recessive parent also, but here we cannot see the presence of recessive parent or the dwarf one. So when we cross the F1 hybrid with the recessive parent, what we are going to see, let's see, it will be capital T, small t, these are the gametes. From here it will be small t, let's fuse it, capital T, small t, capital T, small t, small t, small t, and we have got small t, small t. That means when we are crossing the F1 progeny with recessive parent, we are getting 50% of tall pea plant and dwarf pea plant. And even in the Mendel F2 generation, we have got the ratio of 3:1, where 3 is to be tall and 1 is to be dwarf. So when the F1 progeny used to be heterozygous dominant, then in the F2 generation we will be able to see the dwarf parent. Okay, so by this way we can say that the genotype of F1 progeny, okay, the genotype of F1 progeny is heterozygous tall, and this we can find out by the help of which cross? By the help of test cross, in which the F1 progeny will be crossed with the recessive parent. Clear? So here we have done the solution of the assignment also, and we have also talked about the test cross also.